So today we are going to talk about autoimmunity. And as I said, the first 10 lectures basically form the basis of what clinical pictures that you normally see. And this is one of the cases that you may see in your life, a butterfly rash, malar rash on a young girl, very common in young girls, called SLE. So if you look at the biopsy, if you look at the patient's kidney, because when the problem is, you will see antibody deposition. So there is a test that we do, fluorescent label antibody to human IgG, and you will see that there are deposits of IgG antibody in glomeruli. So that will give you an idea that these are autoimmune diseases, and some of the immune players basically are causing the problem. Another disease you will see, goiter, Hashimoto's thyroiditis, that again is an inflammation, of course, of immune region. And if you were to look at their biopsy, again, you will see over here, this is a normal thyroid, and you see tons of lymphocyte proliferation. Remember, we talked about an immune response where lymphocytes proliferate. If they proliferate, they're uncontrolled, unregulated, so they may end up in thyroid and cause an autoimmune disease. Likewise, if you look at, for example, a diabetes case where you may have some of the typically insulin-dependent diabetes mellitus, you will see that there is a infiltration of lymphocyte. So these are some of the examples that you will see when the immune response goes out of control. So you end up cells where you're not supposed to have cells. And you will end up having antibodies where you're not supposed to have antibodies. And we pretty much need to know how these are regulated and how are we going to cure for them. So autoantibody, just when you thought that it's an auto, so you can see that these are antibodies. If auto is a antigen, so we have an autoantibody. And autoantibody and autoimmunity is taught in perspective of tolerance. So we will, in today's lecture, we'll talk about the same concept that I've been kind of hammering through, self versus non-self, and what are the mechanisms in place. And you will hear me talking about the central tolerance, peripheral tolerance, something we call immune privilege. And then finally, I already mentioned three, and there are more than three diseases that you will see in your clinical practice that unfortunately we may have no cure for, so to speak. So we pretty much know that there are always chances that your body generates self-antigen or something we call autoantigens. So there has to be check and balances between our innate and adaptive immunity. And there were quite a few experiments. Some of the recent studies that if you are interested, you can look it up, especially Peter Medawar's experiment in 1950, and that got him a Nobel Prize. So you can see these are very important concepts you have to have. So let's figure out what is self-tolerance. Self-tolerance, which is desirable, is basically a state of unresponsiveness to self-antigen. We don't want to respond to our antigen. Well, having said that, there has to be a system in place. This is not that easy, so we need to know what happens. If there is an autoantigen and if there is a self-antigen specific lymphocyte that can initiate activation, and we'll end up with all those diseases that we don't want to have. So body has at least two systems in place, two mechanisms. And then again, a whole chapter will talk about this tolerance. Either it's a central tolerance or it is a peripheral tolerance. When we say central tolerance, we basically are talking about thymus, and we are talking about those early stages of thymus maturation. So when T cells are produced in bone marrow, and they are then transported to thymus, and in thymus, they will go through education, so to speak, and they will have their TCR installed. So there is a negative selection in primary lymphoid. Those T cells, when they are made and they happen to react against your own antigen, have to be eliminated. But having said that, there are always alternative mechanisms, or there are always some hypotheses that we do see that happening. And one of, or many of the hypotheses is that some of those lymphocytes may escape there. We miss those cells, and they are sitting somewhere in our body, and then they will come and haunt us later, especially in later part of your life. Now, the question you have to ask yourself is that what is going on in terms of our body to distinguish between self versus non? Do we belong? Does this cell belong to me or not? Because of course, it's a good reason for bodies to have that. Bacteria, viruses, and all these microbes do not belong to us, right? And then cancerous cells do not belong to us. Anything that is not good doesn't belong to us. So what normally happens is that our antigens are seen by our immature lymphocytes, and then they develop a mechanism which is called tolerance. So this is their inbuilt. But what may happen is that if you expose the same antigen to the mature lymphocytes, they may activate. So obviously there are some conditions where this kind of dichotomy, this kind of a particular route has to be taken. What makes them go towards the tolerance and what makes them go towards the activation, because they're two difficult opposite directions. Now also remember that immature lymphocytes, while they are in the thymus, they are more sensitive to strong signals. So they are more sensitive to strong signal, and that basically will lead to a negative selection that we normally see. Okay, and it's both for B and T cell. And then the other important thing that keep in mind is that sometimes what happens is that you, for example, you may have a continuous exposure of the lymphocyte to high antigen level. That is another thing, because some of the lymphocytes can disappear and escape, and they never see an antigen. But then there is another scene where your lymphocytes are seeing that signal over and over again. So that kind of irritates them, and they begin to respond to it. And not only respond, we call it like chronic antigen receptor signaling. So kind of activate it over and over again. In immunological terms, when an antigen is seen by a T cell, right, and if you remember, we have to have a co-stimulation. So if there is no co-stimulation, one of the mechanisms is that if there is no co-stimulation, the lymphocytes will tolerate it. We'll call them tolerized lymphocytes. They would not activate. They have to have two signals. They have to see with two eyes. They have to hold with two hands. So that's pretty much it in simple word. So one of the reasons that there will be tolerization is the absence of co-stimulation. So let's take two separate mechanisms and see what normally happens at the cellular level. That's about central tolerance. There are three different mechanisms proposed. One is energy. If you remember, we talked about that when the cell decides not to respond, we call them energized. They are not going to respond. The other thing we call receptor editing. And then again, if you remember, when receptors are being prepared genetically, you basically edit them. So you pick up different VDJ segments, pick up different genes, and give this editing. And then finally is that maybe there are some clones of cells, they pretty much fall ignorant. We call clonal ignorance. They don't want to react to it. So let's take them in detail. For example, number one, energy. So the whole point for this part over here is that there is a functional inactivation of a lymphocyte. So for some reason, lymphocyte does not respond. So it would not respond whenever it comes with self-antigen. So of course, this is good, right? And then again, if it happens to be a B cell that is anergic, it doesn't want to respond. So obviously, if it's not going to respond, it would not communicate, and there will be no antibody formation. And there will be a growth arrest of at the developmental stage. So all these cells go through developmental stage. If it happens for T cells, for example, if the T cells don't want to respond, so of course they would not proliferate, they will not produce cytokines, and there will be no TCR signaling. And then again, in this case, many of them will be eliminated by negative selection. So these are like simple explanations that we have for one of the first mechanism for central tolerance. The second thing is receptor editing. And again, you will see me talking about B and T cells at the same time. This happens to be the major mechanism in the B cell. Remember, when we talked about antibody formation, we talked about this receptor gene rearrangement. So B cells will always rearrange the receptor genes because they have a choice from V, D, J segment. So they're always a rearrangement. And then if you remember, we talked about recombinase, which was one of the enzymes there that will help the body to pick up and cut and paste. So that's in RAG1 and RAG2. And that also have a role to play in terms of re-expression that you normally see. When it comes to TCR, and if you remember this morning when we talked about TCR hypervariable segment right in the beginning, in the middle of part, so there is a locus rearrangement that basically occurs continuously on both chromosomes until positively selected. So again, you have to understand what is the difference between a positive selection and a negative selection. In both ways, it helps us to provide tolerance to our self-antigen. And this is a little bit different for B and T cell. Now, for the third part, in which if we do, let's say by chance, by mutation, if we happen to have autoreactive B and T cell, so we want to eliminate them. So that's again simple deletion. So just you make a mistake, you delete it. So that basically happens by apoptosis. It's not that we don't have the ability to make mistake. We make mistake. We make those B and T cells that may recognize our body as foreign. So what do we do then? We delete them. And the process of deletion of these autoreactive cell is apoptosis. And you can see if it happens at central level, which is a dominant mechanism, we have negative selection for autoreactive T cell and thymus. So we'll pick up all those cells and just let them die. The other important thing is if those autoreactive lymphocytes have, let's say, escaped for some reason, that tolerant, so there has to be a mechanism to not allow them to attach to your own self. This kind of ability is that you have, we call it clonal ignorance. So we just kind of, even if you see them, we ignore them. Let's say for some reason they've been made, they've been eliminated, and you don't want to respond to them. So body uses something we call clonal ignorance, whereby we deal with those cells which were not negatively eliminated over here in thymus as a central deletion, and now they are in periphery. So what happens is that they will have weak affinity and low concentration for self-antigens. So number one, remember they need to attach to those cells, so they will not really attach tightly, that they will cause problems. So it's a weak affinity. So that's another protective mechanism that you may have to protect yourself if you happen to let those autoreactive B and T cell in your system. Now, that was one protective mechanism. Let's say everything's okay. Most of us, hopefully, will have that process going or already went through. But let's say if there are occasional times when there are autoreactive B and T cell and they are in circulation. And most of the time we see that activity in B cell. So we call it autoreactivity in B cell, and that may occur by a somatic mutation. If you remember, like any genetics, So there's an ability for when the antibodies are picked, gene rearrangement, 1 in million or 1 in 10 million. There may be a lower incidence for that, but there always are chances for mutation. That's why we do see autoimmune diseases. If it's a class of 100, I'm not going to ask you, but well, the incidence will be very low. Right? If, for example, some of the diseases like sickle cell anemia is like 1 in 400 in African American population. It's very high, sickle cell anemia. Right? But otherwise, you have to set loads of people to, so incidence is low. That's the good news. Okay. Now, if we talk of peripheral tolerance as well, again, one of the things is common, energy. And remember, this is just a word. I'm just telling you that cells do not activate, right? But I'm not going in detail what are the mechanisms. So if you are interested, of course, there will be tons of research going on, papers going on. What will make the T cell not to respond? We know for sure they don't respond. We precisely may not know the details for that. The other important thing is that something we call FAS. We call it FAS-mediated activation-induced cell death. FAS basically is a tag. It's just like, you just, in your offices, you just throw it in the garbage or just kill it. So that kind of a tag, you have to put that it needs to be garbage, it needs to be discarded, it needs to be, so that is a tag that cell is attached with. So all those killer cell will come and they identify that, oh, we see that tag, they are FAS. So we call it FAS-mediated activation-induced cell death. So there are different mechanisms for cells to die, right? Because by the end of the day, bacteria, viruses, or cells, we want them to die. We should differentiate by, by a tag that would differentiate that we don't kill ourselves and only kill the intruders. The third important thing, if you remember when we talked about the control of T cell activation, I told you there are important cells we call Tregs. So Tregs are also recently being considered as one of very important players to manifest peripheral tolerance. So keep in mind that if we are treating autoimmune disease disorders, we are working on all these prospective mechanisms, and we are either, let's say, for induction of Treg cells. So we may be preparing Tregs and use Tregs as a treatment module for those particular autoimmune disorders. Now, let's see number one in a figure, and this is a legend of the figure. I just told you earlier, based upon your previous information, that as a rule, lack of co-stimulation will energize B cell. So this means if there's no co-stimulation, they would not kick in. So you have to have two signals, right? Now, in this case, let's go to the picture. Maybe it will give you a better idea. So both of you who are visual. So you can see from here that what is it that called B cell energy. So B cell needs to talk to T cell. We already talked about that in the previous figures. B cell is an antigen presenting cell. It has a first signal. It picks up the information from bacteria, sends a signal in, right? And then talks to a T cell, presents a part of it. And by the end of the day, you want this B cell to produce antibodies. That's a normal immune response, right? So in order for B cells to make an antibody against this bacteria, they have to have this interaction between CD40 and CD40L. This was one of the important co-stimulatory molecules I mentioned this morning. Now you know where is it coming from. So that is considered as a second signal. So if both of the signals are there, you have one provided by this antigen binding and cross-linking of the BCR, the cross-linking over here. And the second is provided by the interaction of CD40 on the T ligand on the T cell. So what will happen is B cell will activate and they will produce antibody. Okay. Now in other scenario, if you look at, for example, B, if there is no second signal, you just have a first signal, there is no second signal, right? So what will happen? B cells will decide energy. They would not respond. So the upshot of that is that we figured out that in order for normal immune response, you have to have a second co-stimulatory signal, right? And now it may look very simple to you, but once we talk of some of the drugs that they are there, you will see that they are going to either bind, block, and do whatever they need to do. In second part, as I said over here, again, you will see that there's a dendritic cell, and it needs to talk to a T cell, and first signal is there. Second signal in this case is B7 to CD28, one of those important co-stimulatory molecules that I said that I will test you for, so you should know that they actually are the proper binding site. So B7 talks to CD24, it is a second signal, and the T cell gets activated, it produces cytokines like IL-2, right? Now, what may happen is that in this case, again, there is a tissue cell, so this is your own cell, and it has a MSC class 2, right? And the T cell will come, and with this interaction, it can get activated and may cause problem, but since there is no co-stimulatory second signal, so T cell will energize. So this means that T cell would not go after your own tissue cell. So that's basically a mechanism whereby your tolerance is produced. And why we are talking about tolerance? Because if you lose tolerance, you have a disease called autoimmunity, autoimmune disease. Now remember, I just told you about the FAS. And FAS basically is, we call it death domain. So this pretty much a sequence of events, very long chain of events. We call them caspase cascade, and ultimately it leads to death. So if you look in a pictorial view, so what you normally see is that one of the cells is expressing the FAS ligand. Now remember, there are some things that I'm not going in detail. What causes that FAS ligand to be expressed? What are the mechanisms? I'm not going in that detail. What I'm just telling you is that once that FAS ligand is expressed, right, then T cells basically will come because they have a corresponding domain for that. So they have a death domain. So they are prepared for that. We call FADD. So FADD basically talks to FAS and then basically will kill the cell. So it's very precise. And then again, when it comes to killing the cell, things are not easy because body goes to a sequence of events. It's very complicated, as I said earlier. It may, and we of course don't want to overburden you to know what those enzymes are there, what those proteins are there. But eventually, as we move along, you will figure out that that's where your drugs are acting. So there are drugs right now in the market, Caspase 1, Caspase 2, so on and so forth. But what they are doing it, and they are working over here. Because remember, sometimes we have to stop cell death, and sometimes we have to induce cell death. If it's a cancer, we want to put this tag on cancer cell. Do you agree? Right? We want to kill cancer cell. And for example, those people who are working on aging, for example, why do you age? Because your cell die. You know, wrinkles, aging. This, so we want to take off that FAS from those cells we don't want to kill. So this goes like a, you know, both ways. So we can. But you can see from here, there is a FAS over here called death domain, and these are series of caspases. So like almost from 1 to whatever, and then eventually will cause DNA fragmentation, and then again T cell. So what happens is that if you want to, this cell may have a potential to go after your own tissues. So we want to eliminate this T cell, right? So maybe in the periphery there is a cell that has a fast ligand, and it will eliminate that or delete that from the circulation. So we don't have those cells circulating that will induce or activate the cascade pathways. You can see these cascade pathways. So those of you, I don't know whether we took apoptosis in detail in pathophysiology, but apoptosis is important because many cells in our body die by a process of apoptosis. So we need to really go and stop that happening. Now the third thing we mentioned was T regulatory cells. I thought that was something important, that we need to have these special cells. And remember, these special cells operate and they take a signal because it's a serious matter, because these diseases are very serious. They're chronic ailments. People have to live all their lives with it. And they are very debilitating, and they are nasty. And we are still struggling to find a disease. And we are at that level. We want to find out what is the immunopathogenesis, what are the immune mechanisms responsible for such disease. So now you will see that some of those co-stimulatory molecules, then when we talked, express on B and T cell, and you were wondering why do we need to know them and why are they important. Now it turns out to be that is those specific co-stimulator molecule that we are after in terms of developing drugs, and in some of the cytokines that are there. Now, if you remember the cytokines lecture, I said there are some cytokines when they act on Th knots, which are undifferentiated T cell. So those cytokines have an ability to lead them towards Th1, Th2, or whatever. So the cytokines are there. When we talked of T regulatory cell, so I told you at that time there are two important cytokines. One was IL-10, you can see over here, and one was TGF beta, right? So tumor growth factor, that's what it stands for. This is a tumor growth factors, because tumors grow because they release those factors. So tumors, cancers release those cytokines, and when there are too many cytokines, they cause death, and we have to stop them, block them, do something for them. Okay? Now again, let's see where is the signal coming from. Again, basic immunology, primary signal, second signal, co-stimulatory molecules, upregulation, downregulation. So if you can really get a good grasp of that B cell activation and T cell activation, you will have easy time when you understand cancer chemotherapy, autoimmune chemotherapy, and many other chemotherapies that are there. It will be very easy for you to remember the action of the drugs, and hopefully you will retain it for a good amount of time. Now, let's see the whole scenario. Now, what's happening over here is that you have a cell. You can see, let's begin from antigen-presenting cell. So there is an antigen-presenting cell, and this cell has MSC class 2 molecule. It's picked up an antigen, and this basically needs to act with a T cell. So there's a T cell is trying to communicate with. Okay? Because by the end of the day, for any immune response, you need activation of T cell, and one of the activation processes will be you need IL-2 to be produced, which is a growth factor. So what happens is that this antigen-presenting cell has co-stimulatory molecule, which is B7. This is the other name for CD86, CD80, and 86 are B7 molecules. So if you look at your original book, it will say that, B7, right? Normally, they should react with CD28. And you can mark it down. I'm pretty sure it's going to be the question. B7 talks to CD28 normally to activate the process. But when the activation goes up, you want to bring it down, you want to suppress it. So what you would do is the competitive inhibition. So instead of CD28, you bring another receptor which is called CTLA4. And that's what you see over here. So CTLA4 then talks to this, and basically what it will do is that it will set the tone as a negative signal. So you can see the block over here. Because this is a tolerance mechanism, you don't want T cells to react to your self-antigens. Either they become energized, or they become deleted, or you have this is a Treg. This is a cell that has many different molecules, and you can see from here. And the most important one is that Treg. The other important thing that you will see over here is that there are lag molecules. I don't want to go in detail, but just remember for your class, I think, because you already know that MSc class 2 talks to TCR, and these two talk to each other. And by the end of the day, it produces those inhibitory cytokines, IL-10 and TGF-beta, that will stop CD4 from making IL-2. So that's the whole sequence of events. So there's a whole cycle, because if there is no IL-2, which is a growth factor, there will be no expansion for T cell. And then your cells, as you saw in the initial pictures, in the nephritis case, a lot of T cells were there causing problems, especially in goiter. So these T cells are being produced and replaced over and over again because lack of this mechanism. Had there been a mechanism like this in place, so the chances are that it would have never happened. Okay. Now, you also remember this morning when I said that we take a lot of antigens, and especially orally. So there has to be some mechanism within our mucosa that our gastrointestinal mucosa normally should not react to everything that we take in. So we have to have something we call oral tolerance. So we have this ability, most of us do. Why do we have it? That's the scientific question. You have to ask yourself, why do we have oral tolerance? Why do we normally do it? So the scientists will give you their hypothesis. This means that hypothesis is a good scientific observation that needs to be either proved or disproved, and you will see cases in both sides. So you can see, because there is a lack of humoral or cellular response to ingested, that's a good news. So food antigen basically do not initiate humoral or cell-mediated responses normally. And then again, most of the responses that you see, especially for T cell responses, they are dependent upon antigen dose. So our antigens in food may not be of that higher number that they can initiate the response. That is another hypothesis or proposed mechanism, so to speak. And then again, there is a low dose. So what happens is that if an antigen is of a low dose, they make another subset of CD4 T cell, we call them Th3. So they are considered as suppressor cells. So they kind of, even if there is inflammation, they will try to cool it down, dampen it down. And then again, of course, you have to have a very high dose to induce T cell energy or deletion. And finally, if you remember, there is we call a very strong immune response in gut. So human gut has the maximum number of lymphocytes. Sometimes we wonder what are they doing there. So we call GALT, gut-associated lymphatic tissues. So there are tons of them. They are in villi, they are intraepithelial lymphocytes, they are in lamina propria, and then they are in Peyer's patches and many different places. So that again is a whole project by itself. For this class, you just need to know that fortunately we do carry good tolerance to oral antigens in terms of food. Now, some other interesting concepts that you may want to know is that some of our sites in the body are immune privileged. So they have like immunity. So this means that they do not develop immune response to pathogen, tumor cells, incompatibility, transplantation. So that is a very, very good news. And no wonder maybe it's a homeostatic mechanism, because you can see at least, I would say, 3 of them are related to the offspring production. So there's maybe a human conservation thing that body has made sure that the reproduction takes place. Because if the reproduction thing would have gone under, if you put that under an immune response, then there will be a lot of problems there. So they have made sure that testes, ovaries, and placenta, they are taken care of most of the time. So we don't need to have that compatibility in terms of reproduction. So that will most of the time. Then again, eye and brain are also known not to have, so we call them as immune privileged sites. So this means normally these would not act as an antigen, and there should not be they should not provoke. But at the same time, this will also give us an insight as to what is happening there. Because as a scientist, that's not enough for us to know why they are immune privileged. We want to know why. And the reason is, if we know why, then we can use the same tools somewhere else, like protecting yourself from autoimmune diseases, protecting or finding a cure for diabetes, so on and so forth. So that's the subject of study, and of course wisely. So we need to find out what are the mechanisms, and we have a lot of. So whenever there's a good scientific question, based upon the data, you have to come up with a hypothesis. You have to come up with a logical reasoning, do experiment, prove or disprove it. That's what it goes in terms of that. Now let's see, what do we have? Well, we think that the reason that we don't have immune response, because they are protected, they don't have lymphatic drainage. That's another thing we are thinking. So the antigens from testes, ovaries, and other places, placenta, are not broken apart and presented to the immune response to test. So that's one of the, and the other thing is they're all protected by barrier. So there's a blood-brain barrier in the, in the brain. There's a blood testicular barrier to protect. So these sperms, eggs are highly protected, and of course for a good reason. The other thing we found out, and that's where most of the research goes on, that they happen to have those cytokines which are immunosuppressive. Remember when I say pro-inflammatory cytokine, this means increased inflammation, increased response, like immune response. But then you have immunosuppressives like steroids. So like steroids, we have this IL-10, TGF-beta, and expression of FAS-L. So these are some of the things that we are learning from that. And we're doing some experiments and figuring out that these basically are some of the mechanisms that we can try, especially for these two players over here that are secreted by the cells, and they stop that activation. Now, just very briefly, I'm going to give you an overview. Hopefully, we'll take it later. Link between autoimmunity and disease. Number one, we do think there is a genetic susceptibility. That's what we think, right? And good reason for that. So if you have a DR3, you may be predisposed to such and such disease. If you're DR4, remember this HLA locus. So if you go for your gene testing, and many people do, and many times it comes in the news that such and such people, celebrities, they want to find out if there is a higher incidence of breast cancer in their family. So if there is an incidence, then they can probably advise as to what to do, what not to do. May it include removal of breast or ovaries and so on and so forth. So that is genetic susceptibility, and each one of us can do it, should do it. That's a discussion in itself. Now, there could be environmental susceptibility as well. That's another issue there. And I'm going to talk about in this one, especially these three important things: release of sequestered antigen, molecular mimicry, and polyclonal activation, because that's something that I want you to understand in terms of what is going on. Now, let's take one at a time. So let's look at A over here. So you can see This one over here is a self-antigen. This is a self-antigen, right? So what normally happens is that the self-antigen is presented to autoreactive B cell, but they are unable to activate because the T cell would not come and help them up, right? Now, in the other scenario, if you have microbial antigen, because remember, we need to deal with microbial antigen. The same machinery is going to work. So if it's going to go after microbial antigen, it can go after you as well, right? So in this case, what's happening is this is a microbial antigen, and the B cell see that, present it to MSC, T cell come, latch on to these two important co-stimulatory molecule, and they begin to produce antibodies, and these antibodies will take care of this microbial antigen. Okay, so this is something that we want, and this is something we don't want. Okay, now if you pay attention over here, is that if there is a cytokine, this is a lipopolysaccharide, which is a component of the cell wall, for example. It's a lipopolysaccharide, and then there are different clones of cells and antibodies which will be generated in response to that. And then these clones will then act. You can see LPS is acting on this, and they will call activation. So you can see that we have an ability to basically activate T cell depending upon the clones of these antibodies. What I want to emphasize in this one is that when you have multiple clones in response to an antigen, and there are tons of clones, it may occur in the process of doing so, you may end up forming an autoreactive clone as well. So you may end up having an autoreactive clone as well. I gave you an example this morning as well, that you want to take control of Streptococcus, and you make antibodies, and antibodies are there basically to take care of this microbe over here. Now there is something we call molecular mimicry. So the molecule of the bacteria carries the same sequence or same epitope as is present in another part of our body, like heart valves. So the antibody which was there to attack that particular bacteria also goes and attacks your valves of the heart, and you will have rheumatic valvular disease. That's the basis of the problem. So whenever you go, very common, especially, and that's also very dangerous as well. Those of us who have kids, they're very careful if it's not strep throat, because if it happens to be a strep throat and you don't treat it, and your body makes an antibodies, those antibodies, of course, will take care of that strep, but will also attach to the heart valves or even kidneys, and they will call kidney damage and heart damage. So that's where the antibodies, by mistake or by design, will pick up the epitope which has, we call molecular or molecular mimicry, same present over there. So that's something that we already discussed that, okay? Now, let's see, we talked about molecular mimicry. What is it? What is it that causes that? The idea, you understand the idea, right? The idea is that there's a good reason for these antibodies to go and attack that bacteria, right? But if you look at the structure of that particular part of the bacteria, on the spin side, what happens is that some of the bacteria, not all, it's just like a jacket, an outer coat they put on, and then come and present to your T cell, the disguise. So that disguise for some of the bacteria can also cause a problem, and then we end up recognizing our own self as foreign and build a very strong response to it, and then we have a problem. Okay, does it make sense? Okay. Now the other thing is that we talk of polyclonal B cell activation, and you saw in earlier as well that normally one B cell would produce only one set of antibodies, right? But some of the bacteria, like the one that have LPS in the cell wall in the previous figure, they basically may provoke more than one kind of response in terms of production or in terms of engaging multiple B cells. So if they hire or engage 5 B cells, so we'll have 5 different types of antibody production. One of those 5 may be the cause for the molecular mimicry that will damage your heart valve. Because the more you stimulate, right? So that's why LPS is considered as a polyclonal activated. So this is very important, a very important cell component of gram-negative bacteria. That's why we struggle to treat gram-negative bacteria. The other thing that you saw, I didn't mention about that, but I did mention it earlier, that LPS, basically, it binds to a receptor on the B cell, which you saw, and it associates with a Toll-like receptor, which is a part of innate immune response. We talked this morning, pattern recognition receptor, which is called TLR4. And why we are talking about that? Because that will be a part of the drugs that now we want to use, not an antibiotic per se, but very specific molecule that will go and help us to treat that. Now, a couple of slides I did mention earlier. There are drugs and hormonal triggers. We saw SLE. Many other drugs are there where they will alter the epitopes of self-antigen. So this morning we were also talking about that normally they should not, but they can trigger that. So that's like we call some of the drugs will trigger it, some of the hormones will trigger as like an autoimmune disease. So in this case, you can see what happens is that penicillin, it binds to proteins on RBC. So penicillin will pick up some of the proteins expressed on RBC and then kill them. So once you give penicillin, you may lose your RBC. You end up having drug-induced hemolytic anemia. So that's caused that. And then again, some other drugs like alpha-methyl dopa, they also cause anemia. Chlorpromazine, hydralazine. So you can see tons of drugs are there which may produce anti-nuclear antibodies. And good thing is that they are being produced. But many times they make mistake, and that's not of a big damage. Friendly fire will take care of it, right? The only thing that you want to do is that if you have a lot of reactivity for these drugs like penicillin, methyl dopa, procainamide, chlorpromazine, hydralazine. So what is happening is that they are generating anti-nuclear antibodies because of the structure. So what you need to do is that if everything is in control, what you would do is that just discontinue the drug, and that should take care of that. Now, this is an example of a activation of a DNA-specific B cell via TLR signaling. So you can see that there is a DNA. So there is a DNA over here, and your cell will recognize that, and then it will produce an antibody. So see the antibody against this DNA. So the whole idea is that once that particular endosome is taken in, it will generate the activation signal, and you will make a double-stranded DNA. So normally DNA would not cause a response, but you can develop autoantibodies against your DNA, and that happens when everything is kind of endocytosed in. So this basically is some of the mechanisms, especially when your cells, specially activated DNA, they begin to secrete antibodies that will target your DNA. And I think we discussed earlier as well. I'll just take a minute or two to give you an example. And this is especially myasthenia gravis. And you have, what is happening is that you have made a anti-acetylcholine receptor antibody. So your body sees your own acetylcholine receptor as foreign, and you make antibodies to it. So you fill in the site, you block the site, and then acetylcholine will not act, and then you will have a disease called myasthenia gravis. Or what may happen is that you have a thyroid follicle and it has those receptors. So these receptors are taken as foreign by your body, and you develop an anti-TSH receptor antibody. So what will happen is that instead of TSH binding to TSH receptor, there's going to be competitive inhibition, or this antibody will bind, and then it will kind of start kicking in thyroid hormone, and that's the basis of Graves' disease. And this is very common. So you can see the whole idea is your receptors are protein. Any structure that your cells see as a foreign, they will react to it and they will make antibodies. And these antibodies will go to bind them. They can either inhibit them, stimulate them, block them, and do whatever. And then this normal function is lost, and then you will have them. Or at the worst, what they will do is that if you make these antibodies in excess or cells in excess, they will go and sit in those particular areas like thyroid, like kidneys, like your islets, pancreatic islets, and will cause immune inflammation and then compromise the function that they may have. Okay?